Hello, everyone. Welcome to this new episode of our Student Spotlight series, where we're able to showcase some of the work that our past students have been doing. I'm Daphne. I'm one of the podcast hosts here at CCIR. And today it's our pleasure to have with us Vanessa. So, why don't you tell us a little bit about yourself, like how old you are, what grade you're in, how long you've been with CCIR, stuff like this?
Hello, my name's Vanessa. I'm right now 15 years old and I'm a grade 10 or sophomore student at the University of Toronto Schools. And I joined CCIR as part of the Future Scholars Program in November 2023. And my course was called Computational Genetics and Drug Discovery, which is like combating infectious disease.
Amazing. And how was your experience with the program? Had you done something similar before, or was this totally new to you?
I think like research, this is my first time doing research, especially with like professors. And the professor that was in charge of my course was Professor Zarkin, and it was absolutely incredible being able to work with him and our teaching assistant, Martha, because I feel like it was such a good experience to be able to have, because it was a small group, right? So it was like a lot of one-on-one time and personalized feedback and advice. So it was definitely very helpful. And I feel like I learned a lot about the research process, which was really daunting before.
Amazing. That sounds great. And could you please tell us a little bit about your topic, like how you kind of chose it and why?
So for my topic, to be honest, this is my first time in like microbiology. Since before I was just like, I've always been interested in biology. So I would like self-study and do like read textbooks. But I was like, I started to like dive more into like neuroscience and stuff. But then I found this. So I decided it'd be really interesting to see what this is about. So, and then when I got into it, I started like reading papers on Staphylococcus aureus, which is like a focus in our course. That's like an infectious bacteria, which is kind of known for being like very geographically isolated with many different strains being prevalent in different areas. So that's kind of what I thought I wanted to compare between like different countries. So my paper is on the comparison of healthcare associated Methicillin Resistant Staphylococcus aureus, or MRSA, because that's quite a mouthful, in Canada and in the United States, specifically New York, against China. And I chose like genomes in the Shanxi province. And so how I got into this, as I said, like MRSA is known for being like very geographically isolated. And there's something I found interesting is that when I was reading papers, there weren't many actual comparisons between countries. And so I decided that's something I wanted to do. And I wanted to choose like very geographically far apart areas. So they could be very different in terms of culture, traditions, but also, most importantly, antibiotic use. And so when I was doing research, I found out that Asian countries, so they're more known for their heavy use of antibiotics. And with many papers that I've read, it showed that like bacteria in those areas are usually more resistant due to that. And they've been able to evolve to the environmental pressures. And then through that, I chose China because it has a lot of data. Like there's so many genomes out there for me to choose from. And yeah, and then so I started finding like the differences in terms of like the genes that they might have, which encode for the resistance.
Wow. And so did you use real data from hospitals, from medical records, things like this? Or how did you collect your data for this project?
This is like one of the most interesting things I found, like how to use like different tools. So I found these all on like NCBI's website. So like for China, it was really easy because like to be honest, because there's a time constraint, I kind of like searched up like different strains, and then I found this paper which was doing a study in like this specific tertiary hospital in the Shanxi province, and it had 38 genomes, which is perfect. And I only chose this one location because there was like a lot to choose from, and because I had trouble finding genomes in Canada and New York, since there's actually not many. And you're probably wondering, why did I choose like it's kind of weird because I chose like a province and then a city and then a country. So what's odd about that is because so China has so many that there's so many for me to choose from. Like I found a lot of different ones, but I settled on this one because there was a lot of them in one location. And for Canada, I searched really hard. I'm a little biased because as a Canadian, but I searched really hard on the internet for a few hours. I could only find 5 genomes. So then I decided to like expand more because I wanted to compare more like Western and Eastern. So I chose New York because it was still relatively close to Canada as well, and in terms of like diversity and such.
Interesting. And was that challenging for you, having to deal with different kind of entities? Like you mentioned, you have one country, or you kind of have two, I guess you have a province in China. So you have one country, one province, one city. Was that like psychologically or like literarily for your paper a bit like tricky for you to manage, or how was that?
It was actually really difficult because I feel like I have a lot of like in my paper, I have like a huge error section because of the time constraint. Like I have like 52 genomes in total, and 38 of them are from China because of like the difficulty. So, but I feel like it was actually okay because the results were like not too bad to deal with. It's just like I have to account for a lot. And I feel like this is like it gave me I feel like this is something I could work on in future papers, and also like raised even more passion for me to maybe possibly continue researching whatever's happening.
Definitely, yeah, I see that. And also, for maybe we should have defined our terms earlier, but for everyone listening who does not know what a genome is, can you please define it for us?
A genome is kind of like all the genetic information in like a certain organism, or in this case a bacteria. So like, and like these are all, you can find these on like online, for example, like databases like NCBI, where they just have like a giant like file you can download of like all the chromosomes and stuff, or like in this case like yeah, just like DNA or genetic information. And because it's really long, like there's a lot of like different tools that you could use to like search for what you're looking for, or like use this information, because let's face it, looking through that giant file would be really time consuming.
Definitely, definitely. I think having to go through all that data is an effort in itself, and then you have to actually write about it and put your own 2 cents in. So it's definitely a kind of juggling act for sure. So like I want to hear a little bit about the structuring of your paper and how you chose to lay out the data for the reader. So can you walk me through that as if I know absolutely nothing about your topic, which I don't? So for all those kind of just getting into science, just getting into the world of genomes and bacteria and medical records and all these kinds of interesting things, can you kind of walk us through how you said what you said and why you actually chose to say it that way?
To be honest, well, let's just start with like structure, because I feel like that's a huge part of a paper. And that was like a giant challenge for me, because schools, like in school, you don't have a lot of opportunities to write research. It's mostly like writing essays, and that's very different. So with like learning structure, it was really helpful with the CCIR course, because like, so for structure, there's like different parts. Let's just start with that first. So there's like, you would have like an abstract, which is like a summary about what the paper is. So like, because papers are really long, you want to have an easy way for the reader to like read through and decide if this is what they want to read through. So you have that, and then you have an introduction, which is more like background information, which is really helpful for me, since this is my first time doing like microbiology as well. So they were very helpful. And then after that, you'd have, for example, like your method, so that they know how you got it. Just as you asked me before, it's an important part to understanding how like the collection and what happened in the process. And then after that, you'd have like your results section, which is like what you analyzed and such. And you can have like different figures that you've made and stuff. And then after that, you'd have a discussion, where it's kind of like discussion's like actually really hard. One of the hardest parts, because I didn't know what would go in there. Because like results is like you kind of just like, what did I find in terms of like research? But discussions were more like you kind of tie everything together. So it's like you can touch upon different things. For example, like I had at the end, I had an error section, but I also had for example, like maybe future research and stuff like that. It's like kind of a blend of different things. And then after that, you'd have like a conclusion and works cited. And the how I wrote this is like, to be honest, because it's actually pretty structured, our professor and teaching assistant gave us like a very like structure, like this is probably what you need to have in there. And then like, because there's a lot of feedback, they were able to like kind of brainstorm with us on what we wanted to include. So for example, for me, because I was comparing between different countries and with resistance, I focused on these things called like AMR genes, which are like, there's many different like genes that like code for a certain resistance, uh, that help like bacteria really rapidly like evolve and like become resistant to different like antibiotics. I focused on that, and I kind of try to like base it off of that. And I first like wrote the introduction, because that's more like background information. So while I was learning about the topic, it'd be perfect to write that while I'm there. And then I like I started my own research and analyzing stuff. And with that, I wrote my like analysis area. And then so that's kind of the order you do things, like introduction, analysis, and then you can go on to your discussion with what you've learned, and you can kind of like go back to everything and write your introduction and conclusion, and then your methods when you're done everything as well.
Definitely, yeah. That I think that structure makes the paper flow more seamlessly, if that makes sense, for sure. And can I ask what your conclusions were for this study? Like, what did you find, and how did you feel that set apart from other research?
So as I mentioned, like I couldn't find a lot of papers online about like actual comparisons between countries, which is really odd because like as I said, MRSA is known for being very geographically isolated. So like even in Canada, if I go between like maybe Toronto and Montreal, there could possibly be like very different like prevalent strains or strains of MRSA that are slightly different from each other because there's a lot of differences within those, which was also seen in like papers I've read. So like with my findings, as I said, I focus on like AMR genes. And with that, I found that there's like three like genes that are most popular. And this is like pretty well known that like MECA, which is like one of the most popular genes that helps like MRSA become that helps MRSA be resistant. And that was also found in my study as BLAZE being the second most prevalent, and then alpha-3. And what I mean by prevalence, like because like so yeah, they were the most prevalent, but and then I used like the data I collected from there, and I made a phylogenetic tree to show like evolutionary history, but also like what countries they were from, and also if they have like this gene or not. And this is where I found some really interesting things that I'm still that I think would be like good for future research because I couldn't find it like all the answers here. And like the main takeaways was that like usually when you think of like strains, you would think that like because it's geographically isolated, like all the strains from Canada, right? You think that they'd all come from like one common ancestor. Let's just say like this is a tree. Like maybe all the strains from Canada would be here, and all the strains from America would be here, and then like maybe all the strains from China would be here, right? Because they're very far apart, and that's what I expected. And when I actually made the tree, something that's interesting is that this is America, this is China, like because China's genes are huge. So this is China, this is America, right? And so like they were very well separated, but the odd thing is for the 5 genomes I found in Canada, they were very spread out. Like they were like there's maybe like America, China, there's maybe one here, one here, one here, one here, and like one here. It was really odd because I expected them to all be together, right? And I think a possible explanation for this could like be maybe like immigration, because Canada is known for immigration and it has a lot of movement from like China, for example. But that's really odd because New York is pretty diverse as well, right? And the other thing I found out from this was like with the 3 AMR genes I was talking about, percentage wise, like the 9 genomes from New York, they had the highest presence. So for example, like I said, MECA, BLAZE, and alpha-3, right? With MECA and BLAZE, all of the genomes had it, like 100%. All 9 genomes had this gene. But with alpha-3, it had like a little over 50%. And that's really high because the averages I got for like my study in total with all the 52 genes, like they were all lower than the averages that I got for the New York genomes. And what I find really odd about this is because I expected this result for China. As I said before, China has like high usage of antibiotics, right? So that there's a lot of like environmental pressures for the bacteria to evolve. And rather like the like percent averages I got for China were a lot lower than for the United States. And that kind of goes against like a lot of other studies that I've read. And it makes me wonder if maybe there are maybe like other prevalent genes that are at play, and maybe the ones in China and Asia, maybe there's like other ones that are popular there. So I feel like my research has just driven a lot more questions for me to continue with.
That's absolutely fascinating. No, definitely. I really like the tree analogy you gave us with in terms of definitely. Can you talk a bit more about? I quite like how you kind of explained what you expected and then what you found and how that differed. Can you talk more a little bit about that tree and how the distinctions were going to be very kind of set, like boom, China, Canada, New York, kind of like this, and you found they were more fluid. You kind of clarify that tree a bit more for us so we can really kind of pinpoint exactly why that's significant and why you hadn't expected that and are surprised to find that.
So with my research, like I'll just say this, like first, like it's really hard to visualize. So I use like this I use like the software that was introduced to us in the course to make like a phylogenetic tree which shows like the evolutionary history. And when I did that, like it and then I like labeled the tree with all these like different countries and stuff. And then when I did that, what I found was like it's what I found with this tree was that in terms of evolutionary history, like because as I explained before, like there's like a lot of because it's like a very geographically isolated like type of bacteria, it's really hard for like faraway places to have similarities because it just somehow doesn't spread that far. It's very like put in place, like very local. And what I mean by the results, as in like as you described, it's like in a way there's like a bit of fluid. There's like a fluid movement, and what I mean by that is like somehow like because like let's just say like the middle is like a common ancestor that all of them have spread out upon. So it was kind of what I expected in the way that like from this common ancestor, there's like one ancestor that spread out this way and like evolved to all the genomes I found in New York. And that was like separate in its own entity. Like there was no other ancestors. It was all just the genomes I found from New York. And then the rest of them kind of spread out and diversified to the ones that I found in China. And then throughout the ones in China, because like there's like also like different little clades there's like one branch and it becomes like it branches out even more and even more and even more to all these like tiny little branches. Within these tiny branches, somehow like or like within these big branches that spread out, somehow they separate the Canadian genome. So like you'd expect that it'd all be like China, and there'd be like after like they branch out, you'd expect them to only have like Chinese genomes. So I found it really strange that like there were like Canadians genomes like spread out here and there, and there's like not really an explanation for how they got there or how they became this similar.
That makes a lot of sense. That's very clear. Thank you for that. Yeah. Why do you? This is a very abstract and tricky question. Why do you?
The possible reason for why this happened is due to like I think maybe like possibly immigration or possibly like movement of people. To be honest, I'm not too sure about this, which is, as you said, it's quite hard. But I'm wondering like if I were to speculate, maybe like immigration, because as I said before, Canada is like quite known for being accepting of like many people coming in, and there's like a huge population of Chinese Canadians who possibly like maybe immigrated here, and it could have brought, I don't know, like maybe genomes, because there's a lot of like population movement as well. At the same time, as I said before, I actually don't know why, because like New York is actually a pretty like really diverse place as a big metropolitan city, right? So there's probably a lot of immigration there as well. So it's really interesting to me.
I have one question for you. Do you have any tips and tricks for anyone who's starting a research work?
Any tips and tricks for research?
Yeah.
A huge tip is like, to be honest, I still have no clue what I'm doing. When I was doing this whole thing, I was really confused because it was like completely new, first of all. And I just always viewed research as such like a huge gap in terms of like a learning experience. And the biggest tip or like that helped me was one is to just like not be overwhelmed. Like I know it's really hard when you first get started. And I kind of like this happens every time, like according to our teaching assistant, like every time she warns people to like get started early on their research. But I feel like, and it always backfires because I think we're all a little too intimidated. So I would just say that like don't be afraid of just like following along with whatever the professor's saying or like whatever tools they're giving out, because it seems very abstract now, but let's just say they give you like a random tool. You could just like test it out in your free time, even if you don't know what you're doing, because that's what happened like with a lot of us. Like I had no clue what the things they were talking about were. And I was like, I don't know how this is going to help me with my research. And in the end, it played like such a pivotal role. So I think it's just like to not be afraid and just try things out and just keep going with it.
Definitely. Thank you so much. That's really so much. Well, Vanessa, it's so lovely to speak to you today. I learned a lot, so never everyone listening did too as well. And I cannot wait to read this research paper. So thanks so much for joining us today.
Thank you.